Good evening. Tonight we're taking a closer look at a scientific breakthrough that could change our very own DNA. Scientists are calling this tool CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats and is a gene editing tool that allows scientists to make precise changes to the DNA of living organisms. Supporters of CRISPR say that this technology could cure some of the world's worst diseases, but critics are questioning the ethical implications and consequences it could have. You may be wondering, how does CRISPR work? Well, the mechanism of CRISPR genome editing contains 3 steps: recognition, cleavage, and repair. Scientists design a custom guide RNA molecule that matches the target DNA sequence they want to edit. The designed RNA recognizes the target sequence in the gene of interest through a complementary base pair. The Cas9 nucleus makes double-stranded breaks, essentially acting as a pair of molecular scissors. Then the double-stranded break is repaired by either non-homologous end joining or homology-directed repair cellular mechanisms. CRISPR can be used to delete, alter, or add genetic material. This genetic breakthrough has the potential to treat and cure a variety of genetic diseases such as sickle cell disease or muscle dystrophy. Duchenne muscular dystrophy is a deadly inherited disease caused by a mutation on the X chromosome leading to severe muscle weaknesses. This disease is caused by a variety of different mutations, mainly deletions at 68%, point mutations at 20%, and duplications at 11% in the DMD gene, known as the largest human gene. Most patients die in young adulthood, but CRISPR can make permanent corrections to the faulty gene to make it functional again. CRISPR is not only capable of curing genetic diseases, it can also genetically modify agriculture, which can get rid of crop diseases and make them climate resilient. Researchers are using CRISPR to precisely alter plant DNA sequences known to be related to particular traits in order to breed plants with desired traits. CRISPR can create crops that are resistant to viruses, fungi, and bacteria, reducing the need for chemical pesticides. It can also alter the plant's ability to withstand extreme weather conditions like drought, heat, and salinity. Compared to previous breeding techniques, CRISPR is precise and faster. This could ultimately increase the world's food supply. If CRISPR has the ability to be so beneficial, why are there so many people against it? CRISPR isn't technically a new technology as its discovery and basic principles are over a decade old, but the application of CRISPR is currently becoming more widespread and rapidly advancing. People are concerned about the unintended effects that may come with gene editing techniques. There has been limited testing on humans, so long-term effects are unknown. There are also many regulation challenges that come with gene editing. Another large concern people have is the creation of designer babies, which are babies that have had their DNA edited for reasons other than disease prevention. Equity issues are also a complex issue that comes with CRISPR technology. How will it be determined who qualifies for this type of procedure? These concerns have sparked many online debates about the future of CRISPR. We've pulled some examples online. On the left is a meme from Instagram joking about the good and bad pathways CRISPR could take scientists. On the right is a Reddit post where the user is questioning if CRISPR will cause issues with eugenics. In 2018, scientist Jiankui announced that he had produced the world's first CRISPR-edited babies in China. To do this, he recruited couples where the father was infected with HIV and the mother was not. He had modified a key gene in a number of human embryos in a way thought to create resistance to HIV. This experiment occurred during a time when gene editing was banned in China. He was placed in jail for 3 years and fined for illegal medical practice. This medical malpractice brought up many ethical concerns against genetic interventions. Today, after previous controversy and unethical testing with gene editing babies, scientists are finally starting basic research on gene editing and human reproduction. Scientists are eager to push this technology due to declining birth rates and to eradicate serious genetic diseases. Currently, U.S. regulations prohibit editing genes in embryos that could become babies, but that could change. The Manhattan Project is planning to explore gene editing by working on mice before moving to primates and then human cells, and ultimately working with human embryos. Their goal is to produce enough evidence to fund research and inform the public what technology is capable of. With that said, CRISPR is a powerful tool that allows scientists to fix problems within DNA. It is a technology that offers a way to treat diseases and improve agriculture. I remember watching a video about CRISPR in my college biology class when we were covering genetically modified organisms in our genes to proteins unit. I think that this technology will prove to be useful as it advances, but we have to hope that it is used responsibly. We'll continue to follow this story as more details emerge.